Hello.
Well,
How are you?
I'm pretty good. I have my new glasses right here, but apparently I can see better without them
Oh,
on the computer screen.
oh yeah,
Like,
yeah.
yeah, it's is this better? Is that better? That's better.
Yeah.
Then this is more blurred. I don't know. I'm not sure.
Yeah. Well, good. I mean, well, glad that you got your eyes taken care of for now. And
Yeah. So hopefully I can do some work.
yeah.
You don't know much about signal processing, do you?
Not
Right.
really. It's not my main area. What were you studying or?
Well, optical coherence tomography, of course,
Yeah.
and the formulas around sweat source optical coherence tomography, and there's some probability terms in it. So I'm having some difficulties trying to understand the probability terms. Just
Okay.
yeah,
Yeah, I don't know that.
I'll just shake my head and continue. I guess that would be it.
Yeah, I guess. I mean, it's pretty tough. I mean, like, uh, well, signal processing is generally pretty tough math, so I don't know.
It's mostly just the Fourier transform, which is I understand it kind of,
Yeah.
but to try to prove things
Right.
in it, I
Yeah.
don't know. Anyways,
Yeah. So yeah, we got well, Madhuri's here. She's from our she joined our orthogonal lab group, and she's going to join this group as well, I guess, or she's coming to this meeting. So how are you, Madhuri?
Hi, I'm good. Jesse suggested that I could be a part of this, and yeah.
Yeah, that would be great. Um, Welcome to the meeting, and
Thank you.
Akshay's here as well.
Hello.
Hello. All right. So welcome to the meeting. People will come and go as usual. So we'll see. We'll welcome people as they come in or whatever. So first thing I wanted to talk about today was I wanted to get on. Well, does anyone? First of all, does anyone have anything they wanted to talk about before we start? Any announcements or?
Nothing new. I'm swamped with exams, assignments.
Yeah. Okay.
Semester's getting tough. Yeah,
Yeah.
not able to do work.
Yeah. Yeah. Madhuri, why don't you introduce yourself to this group?
Yeah, hi. I'm Madhuri, and I recently joined Orthogonal Labs, and I hope to start working um, on a project soon as a research assistant. Yep. I graduated with a master's in molecular biology and genetics last year, and I've been a part of two small projects, and yeah.
Okay, so you're used to like a lot of the what they call wet lab work, which is working with buffers and other like actual biological samples, right?
Right, that's
Okay,
right.
yeah. So yeah, this group we kind of mix the two. We have like the sort of the biological side, and then we have the computational side. So that's what we try to bridge here. And so if you have any things you want to follow up on in a meeting, well, let me know or let any if someone else is presenting, let them know what you find interesting. Because usually like the Saturday meetings, there are a lot of topics that we kind of gloss over, And then there's, there are a lot of things we could explore in depth, but we don't necessarily do that because we don't have time. So I find it, I find it useful to go through a lot of these things at a high level because it introduces people to a lot of things, and then you can do those, follow up on those things later.
Right, yeah.
So we also have Jesse and Sanjay. Welcome. Okay. So the first thing I wanted to talk about was, let's see. Well, I did want to bring up the fact that we have Hacktoberfest coming up at the end of the week starts. So I think the first is Friday. So it's going to start Friday. And I still need to get things ready for that. I'm going to share my screen. So one of the areas that we're going to have as a repository for Hacktoberfest is the digital Basel area. And I've been working with Ujwal Singh and Asmit Singh and Tarun and, those guys from the Monday meetings. And we're meeting kind of separately to talk about these, sort of doing it a second pass at the digital Basel area stuff. So this is the project where we have this diatom colony that we're, looking at the morphology, we're looking at the behavior. And so we're using movies to segment out or to analyze those images to define the cells themselves and then define the colonies and the movement and all that. So we have a bunch of different repositories. And of course, we've put out a paper on this, which is a recent book chapter. And it, involves deep learning and biomechanics and all this various ways of sort of characterizing this digital Basel area. So this is the Basel area, of course. This is the diatom. For new people who are new here, they have a number of modes of movement, different types of, this colony sort of flexes back and forth like an accordion, but it also has other modes of movement like quiescence and, where the mode where they're all bunched up together. And sometimes these colonies break apart. And so we have a lot of movies that have been collected by a collaborator, and they're going to look into different ways of analyzing these movies. And so we had some interesting talk about some cutting-edge deep learning technology, and hopefully if they can get it working, they can present on it in this group. They used a pre-trained model for this version of the analysis, which is DeepLab version 3, and that's something you can implement in TensorFlow. And so we have a lot of stuff that we did for this, and we're trying to move to a second phase. And I'm still trying to figure out what the issues are for this. I'm gonna maybe go through this repository and kind of pull out some issues. We also have the Devolern repository, and that's here. So this is the Devolern repository, and of course this has been worked on by Mayuk and Mainak Deb, and they've worked on a bunch. So this has a more active set of. This was active last October, Hacktoberfest, and it's been active all year in terms of accepting contributor requests, or contributor contributions. So if you go, I don't know there's our issues board here. So we have like 6 issues open right now, and we're going to try to create a couple more issues for people to try to address. And again, the process is if you want to contribute, you pick an issue, you fulfill the issue, and then you issue a pull request, and then one of us will accept the pull request, maybe with some modifications. And then if you do, I think 5 contributions. The rule of Hacktoberfest is 5 contributions, and you get some swag. But we don't have that in our group. We just basically, if you make 1 contribution, we make we recognize it. We put your name on a list and we advertise it. In various places. So, and then of course, if you make a number of contributions, of course, that might lead to other things. So we don't go the swag route on this, but nevertheless, we're gonna see how many people we can get in during Hacktoberfest. I'm gonna start advertising for it, so be on the lookout for some advertisements on that. Okay, so next thing I want to talk about is our submissions document, and this has not been updated too much since the last time we saw this, but we have a number of different things that are open. So we have a lot of things here that are closed or that have already been done. We have this mathematics of DevoWorm here, which is this collection of different mathematical things that we use in the group, and right now it's in poster form, and we're trying to work it into some sort of book, maybe for Wormbook or some other resource. So this is still outstanding. I need to revisit this Devolern paper, which has been put on hold for a long time because we've had other things come up. But this is, of course, the thing I just showed you, the Devolern repository. Now, we just finished Google Summer of Code, so that means that we're pretty much at a stopping point for modifying the platform. And then soon there will be a new release on that, so this paper will be active once again. We have some other things, some other ideas like Williamson symbiosis, molecular level simulations of the diatoms that we showed before. This Williamson symbiosis is this idea that some organisms have taken basically the genomes of two different animals, two different animals' developmental programs into their genomes, and they express them. This is for organisms like ones that undergo metamorphosis and other forms of transformation and development. And so there are ways to look at this. It involves a lot of genomics and similarity analysis. So this is something you'd have to be pretty advanced in bioinformatics to do this. This molecular level simulations of diatoms, that's maybe a little bit different. This is where you're looking at motion, you're looking at higher order motion. So this is something that might require us to finish up this analysis of the movies, but it's open. We have another diatom movement item here, looking at the smoothness or jerkiness. This is kind of the same thing. Jerkiness is like where you get really high, really fast movements. So you have these derivatives of position. You have motion, derivatives of motion actually. You have acceleration. You have and then you have higher order derivatives, and those are basically what we'd be looking at in this. This quantitative comparison of Archean shaped droplets. I think this is something that Mynock was looking into. He did a demo a couple weeks ago, and I don't know what the state of that is. So I can actually probably make a note of that if I haven't already. Oh yeah, I did already. Okay. Then we have the NeurIPS workshops. Those are, I think, closed now for submissions. But, if you're interested in attending NeurIPS, if you're interested in machine learning or deep learning, that's a great place to go for that. Just participate. You can participate virtually, I think, this year. And it's a great place to go, especially the workshops, where they have workshops in a number of topics. Not all of them are, just kind of like self-contained machine learning. Some are sort of like metacognition and things like that. So this link to the workshop, let me make a copy of this link here, and I'll put it in the chat for people so they can look those over. And I think the registration for NeurIPS is rather inexpensive. And then, so that's NeurIPS. And then if you're also interested in even more conference participation, there is NeuroMatch 4.0, which is October 25th is this submission deadline for abstracts. So we're going to be participating in this conference. This is a virtual conference that occurs in December, right around the same time as NeurIPS, but it doesn't overlap with NeurIPS because I think a lot of the same people go to both, so that won't overlap too much. But it's going to be a pretty good time. It's usually a pretty good conference. And speaking of which, I've created an abstract submissions document here for NeuroMatch. So, like I said, I have a group on Saturdays that also is going to be participating in NeuroMatch. So what I'm doing here is I'm trying to assemble a list of submissions that we can that people might want to submit, and these are at the level of an abstract right now. So the idea is, if you have an idea you want to think about submitting for, you put it into this list under title and ideas. So this is a poster that we presented at the Networks conference at the Network Neuroscience satellite of that conference. And so we have that already to go. We have a poster of it. So what we would do, we'd say, okay, we want to pursue this. Let's put together an abstract that is, according to the standards of the NeuroMatch submission system. Then we put that in and we hit the submit button, and then if we see this column here where it says submitted, we'll say yes, and then we'll put a link to the abstract, maybe in the Google Doc where we have it. And then we have to follow up. So the next columns would be, do we have the slides ready? The next column might be, has this been presented? Next column might be like notes. What do we need to do to get this done? So there are all these things that we need to put together. Actually, I should put the notes now, since what will happen is people need to be able to kind of meet some deadlines and things before they submit their abstract. So this is if you have an issue with submitting the abstract. So we have this AN and BNN follow-up. This is something we did a presentation on last year at NeuroMatch, or at the last NeuroMatch. And that, again, we can submit a follow-up to that. I don't like to submit the same talk, in subsequent conferences of the same series, because I always think we have something new to say. I'd always like to think that we can add some value to it as we resubmit them. So I would like to, if we do submit something like this, I would like to update it a bit. This same goes for this. It's a poster. We'd probably submit it as a talk, so it would change in it's the way it's presented. But then we could add some new things in as well. So these are, and then there's this embryo connectome networks, which is also a DevoWorm thing, and that's something that's been submitted to other conferences, but in different forms. And so we can recycle most of the slides, but we do want to add something new. So if you have an interest in this, if you want to suggest things that we might submit, please take a look. I think the sharing is on that document. You might want to, if it's not, you might want to request access. But if you want to take a look and maybe put some ideas in, if you have something, and we can follow up on it as we go through, that would be good. I think that's something that is of benefit to most people. Some of you haven't been in the group that much, but that's okay. You can, you can still maybe propose something or work on something with someone else. If you want to work on a presentation with someone else, you could make a note and say this person is interested, and then we can see it through. And the important part here is seeing something through. So you put an idea down and you kind of try to flesh it out over, well, in this case it would be a couple weeks. We have like maybe three and a half weeks by the until the abstract deadline. And then we'd basically work out an abstract, and then we'd have to work out the slides. And then once that's done, you have something to present at a conference. And it's pretty simple. It's like a 15 to 20 minute session. You would go in and you would you might even be able to record it. We would go over best practices for creating slides, which is basically that there's a lot of, it's pretty visual. You want to be fairly visual. You want to be succinct. You want to lay out some of the main ideas. You want to walk through some other ideas, have a conclusion, and then always open the door for future work. So what do I do next? So in the audience there is, it's neuroscience, but there's also some biology. There's some not really a developmental biology track, but there's some there's some interest in development. So that's something we can sort of address. And then there's also the deep learning and machine learning side of things. So if people are interested in deep learning and machine learning submissions, NeurIPS is actually quite competitive, so this might be an alternative to that. Okay, so that's what's going on in terms of submissions and Hacktoberfest. And again, I will advertise Hacktoberfest closer to the beginning of the month, and it goes on for the entire month of October. And maybe we'll get some people to come in and contribute. Maybe I was even thinking about using this group, the meetings, as sort of a Hacktoberfest issue. So attend the meetings, maybe present some idea in the meetings or participate in some way. And that's sort of equivalent to doing some sort of pull request. But I don't know. That might actually not be a bad idea, actually. Some people might prefer that. So, well, more on that later. Let me know if you have ideas about that. You can share them in the Slack. Okay, so now that we've done that, I want to talk about this. Actually, I don't know if I want to go over major tasks. I do want to talk about the OpenWorm general meeting. So tomorrow morning, we're going to have, at least my time, we're going to have an annual update to the OpenWorm Foundation general meeting. And so this is it's like a 5 to 10 minute presentation on what's been going on in the DevoWorm group. So this is the DevoWorm group. This is our web presence. We have a website, a Weebly website, and then we have this GitHub repository, which is where we have the main DevoWorm, the secondary data and a lot of the other repositories. We do have the Devolern repository, which I showed recently here. And this is sort of a quasi-independent thing where, we have this Devolern software. We have some other things that we're working on, model organisms, data science demos, theory building, some other educational initiatives, things that, are kind of in different states. They're not as developed as Devolern, but that's the idea there, is to have an educational portal for people to go through. So this is the talk here, this update for 2021. And in going through this, I noticed that there were a lot of things that we did, and I just didn't know, the pace of things is you kind of go week after week, maybe monthly, you take an assessment of where you are and you just work on things. But it's, going back over the course of a year, you get to see the full extent of what has been going on. And it turns out that actually we've done a lot of stuff in this group over the course of the year. So I'm proud of that. And I'm going to talk about this to the group tomorrow morning. So this is the title slide here. This is the logo for the group. The very arty kind of embryo type logo. So one of the things we do in the group, and I'm going to stress, is deep learning and quantitative morphology. So this includes things like axolotl brain development. You can see that here. You also do C. elegans embryogenesis, which you can see here. There's also this theory building to explain developmental processes, which is this GIF of a sphere turning into a cow. So there's this thing that people use in modeling, and there's also a joke about it, about spherical cows in a vacuum. And so that's what this refers to. Oftentimes when you have an underspecified model and you use it to analyze some really complex system, people often say that you're looking at a spherical cow in a vacuum. And so that's where this comes from. And it's just kind of an idea that, in modeling, we're not doing, we have to really work hard to get a good representation of what we actually want to study. It just doesn't come through like the application of a general model. So we have to really kind of do a lot of work on that. With our embryogenesis stuff, this is, C. elegans embryogenesis is relatively simple in terms of segmenting the cells. But then understanding like their movement and all this, movement, their migration and their division and what they sort of differentiate into, that's a different story. This work here just shows an example of, segmenting a bunch of cells as you go through a stack of microscopy images from the top to the bottom of the embryo. So you can see how this algorithm is able to pick out the cells as it moves through the stack. And so, it can pick out different variations in the morphology. But then you also have this situation where the cells migrate in development, and they differentiate, and they divide. And so all those things have to be captured. And so that's where it becomes, more and more increasingly difficult as you add those things in. And then finally, we have bacillary cell colony morphology, which I talked about earlier in the meeting. So this is telling the larger group about Devolern. This has been a success. It's just hit, this was from, when was this? This wasn't too long ago. It was like a month ago or something. In any case, so our PyP instance of this just hit 10,000 downloads. I think that was about a month ago, so it was up for about a year, and it was about a year since it's been on PyP first. And so it's been just over a year, maybe at 10,000 downloads, so that's pretty good. With over 800 downloads in the last 30 days, which is a good trend to have. If you can get more downloads in a shorter period of time like that, it means people are starting to check it out, and certainly we've made progress on it. When it was first released, there was some buzz, and then it was like, we had to wait a couple months for people to start contributing. And then, now that we have a second Summer of Code project that focused on this, I think we're hitting maybe some sort of critical mass. And so this is a nice advance for this group, and I want to make everyone else in OpenWorm aware, because I know in OpenWorm they do a lot of things. They do they look at mostly at the adult C. elegans, and they're looking at things like worm movement. They're looking at things like modeling the nervous system, modeling different neurons. They're looking at things like the biophysics of movement. So they're looking at things that are not only in the adult, but also have a lot of complexity to them. And, this kind of model might help. I know there's been a lot of interest in machine learning and deep learning more generally in OpenWorm, but we don't really have a lot of people doing anything outside of DevoWorm in that area. It's been largely just other types of models. So this is something that I want to stress. I've had some conversations about education and educational initiatives in OpenWorm, like things like teaching, what they call upskilling, which is where you teach people new skills and it helps them, gain new skills that maybe they wouldn't be able to get in a degree setting, or maybe even like, in a LinkedIn course or some sort of MOOC where they just learn the theory of it, or they kind of learn like from some examples. In this case, you can actually work with the data. So this is our Devolern platform over the 20 and 21 seasons. So we have this umbrella, this Devolern umbrella. It includes Devolern, and actually this should be 0.3.0. Then we have these species-specific models. This is DevoWorm AI. This is like a series of models that are other types of models. We have an RNN that's used for analyzing embryos, and we also have another model for specialized for like the basilaria cells, or those long rod-shaped cells. So there are different tools for different types of cells and different types of developmental context. So we have those, and then we have the DevoZoo, which is a focus on model organisms and digital organisms. and sort of, introducing those. And I think that's something that we can revisit, because I know that, I think I said this last week, maybe it was at my other meeting, where there's this interest in model organisms, not as like what they can do, but like kind of bridging between different model organisms. So for example, the DevoZoo features a lot of different model organisms like zebrafish, fruit flies, C. elegans, of course, spiders, octopi, and then you have these digital organisms that you can create that, allow you to model different aspects of development. So those are all in the DevoZoo. And I think people have an interest in that. Like there's some sort of synthesis there, I think, as well, where we can understand a lot about general phenomena by looking at cross-model organisms, and, having a resource where people can go and learn more about why do we use zebrafish, or what's the utility of using Xenopus, which is the frog model. What's the utility of that? So there's lots to develop on that end. I don't know exactly how to drive this forward in terms of funding or projects, but I mean, I guess the best way to do it is just to, maybe that's something else we could raise during Hacktoberfest, not as a whole, repository, but as an issue. Like look into DevoZoo and suggest changes. So this is something that can be done as well. So then we have these conference presentations and papers, which I want to highlight for the larger group. So I've personally done a lot of work trying to promote Devolern. I did this at INCF, the INCF assembly, which happened in May. And this is something where I presented on Devolern to that group. And it was a really nice, it was virtual. You had a lot of people from all throughout INCF. So there are a lot of people interested in different aspects of neuroscience. They work on this thing called FAIR neuroscience, which is, it's like this acronym for, what we should expect of open data. So, it's like transparency and reusability and all these other things. I don't remember exactly what the acronym means right now, but so we were participated in the INCF assembly with the rest of the OpenWorm Foundation. We did this poster at the International C. elegans Conference on the OpenWorm project. DevoWorm had a little space on the poster, and that was where we highlighted some of the things that were going on in the last year or so. I think I showed this in a meeting, the draft of this poster, but it was actually well received. People were interested in it. We also participated in the NeuroMatch conference, and this was NeuroMatch 3, which was back in, I think, October or November of last year. And we did a couple of talks there. We did this thing, the psychophysics of non-neuronal cognition, which we just finished a paper on this in the group. And so there's a whole paper on this now, and it's coming out as a preprint soon. But you can read about it, or, this talk is online. And this is kind of laying out sort of this idea of that you can use this metaphor of cognition as a way to look at. In this case, we use the bacillary example, but I think this talk was a bit broader. And then we have this contrast between biological and artificial neural networks, which is something that Krishna, Katyal, and myself worked on. So, we participated in that. We also participated in Networks 2021. We did this embryo networks presentation, which is this idea that embryo cells are connected, can form what they call networks, complex networks. So they have linkages, they have, based on their spatial position and their distance, and you can, it's informative in a number of ways. And in this particular talk, we talked about how you can use this type of model to look at what happens as the connectome starts to form and it starts to diverge from the other cells in the embryo. So you have other cells in the embryo that will go on to form other organs. But you do have a core connectome. And the idea is this network will differentiate accordingly. So when you get a connectome form, when you get neurons that differentiate, they form a subnetwork. And those two networks interact. but they're also divergent because they're no longer sort of the same network. They have functional differences. There was also this talk at Dynamics Days on game theory of developmental processes. That's also on our YouTube channel. And then this advanced computational neuroscience network talk is also on our YouTube channel, I believe. This is another version of this networks talk. It actually features a little bit more work on the network side of it. And so that's something you also check out on YouTube. There were also a couple papers this year. We had the neuroinformatics contribution. This was in the special issue, Building the Neurocommons. This is a paper called Data Theoretical Synthesis of the Early Developmental Process. This is something I think I showed to the group at the beginning of this calendar year. This was a paper on, looking at using secondary data to build models and hypotheses, and then also building theory. And there's this whole special issue. This Building the Neurocommons special issue is put out by people in the INCF working group on open data. So they have, if you go to the special issue, they talk a lot about some of the FAIR standards and things like that. So that's something to check out. And I know it'll be a great interest to the DevoWorm or the OpenWorm Foundation, because one of the founders of the OpenWorm Foundation actually has pretty strong ties to that world of neuroinformatics. So, and then there's this biosystems paper with Ojwal and Jesse as co-authors with me, periodicity in the embryo, and this is a sort of a very low level. So I know Susan was talking at the beginning of the meeting about signal processing. There wasn't a lot of signal processing in here per se, but we did look at different aspects of the time series of periodicity of cell division and cell differentiation, and what that says about how order is formed in the embryo. So the idea here is that, I kind of want to highlight our process for open involvement. And so this is a preparation for Google Summer of Code, but this kind of applies year round in terms of people coming into the group. So, you find a call for involvement. It could be a Google Summer of Code project. It could be a set of issues on GitHub. You contribute to a GitHub issue during your application period or anytime. You join the weekly meetings, get involved in bigger projects. So this is something I want to highlight because we talked about education, and I think this could be a very successful model if we could support it and make sure that the foundation is behind it in some way. I have a list of open source contributors. These are just people who contributed to the Devolern repository. So not just the software, but the entire repository here. So we have quite a few people on our list here, and this is not exhaustive. I think I picked people who have put in some contribution to that, just that repository. But of course we have other people who make meetings, and I didn't put them in this list. This was, I think, for Hacktoberfest of last year. So this year, I think if you contribute in some way, you'll make a new list. There are also cross-disciplinary pathways. So we have, and this is something that Madhuri is experiencing currently. This is where we have this orthogonal research and education lab, which I operate as head scientist, and we have Jesse and Madhuri are part of that. And then I think Krishna is also part of it, and people like Shruti who have contributed to this. This is a broader set of opportunities in an area called neuro AI. There's also ethics and computational modeling work that goes on here. And then, you can interact with DevoWorm, which is computational developmental biology. We also are interested in, we talked on Saturday about neural embryoids or neural organoids. And so those things are possible as well. We kind of have this crossover where we have spillover research or spillover opportunities. So we want to kind of make that more explicit. I want to bring that to the attention of the foundation as well. And then finally, thanks to our contributors. So this is not like I said, not an exhaustive list. This is sort of people who are oftentimes regulars in our meetings. So if I've missed you here, I could probably add you, but this is just a sampling here. And then we have our Google Summer of Code people, and thank you for that. So that's what I wanted to cover in that talk. I don't want to. I'm not going to be that far-ranging when I present it to the foundation. I just want to go over some of those points. Does anyone have any thoughts about that, or in terms of future directions?
It looks good.
Thanks. Yeah, so one of the things we've kind of talked about, and it kind of fell by the wayside, I think it was last year, after last year's meeting, was this idea of upskilling. So the idea that you can, interact with a group that will allow you to gain skills in some area. So, people are often, like machine learning, that whole revolution has happened, say, in the last 10 years, maybe 15. And imagine if you started the workforce back in like the early 90s and you had an undergraduate degree and then you didn't go back to school, and you don't really have time to go back to school, and so you need to learn this new skill. So that's one thing you can learn is that's upskilling in terms of new technology that you can learn, and there have to be pathways for that. Another option is that I've had requests from other people in OpenWorm who are computationally oriented on learning about microscopy or wet lab techniques. So that's a little harder to do virtually. But that's another example of upskilling. People don't necessarily, you can't just, walk into a store and say, I want some wet lab skills. You can take a course at a local college, but it takes time to do that. Like you have to commit to an entire semester, pay a lot of money for it, or, so, and then you still may not really get the full experience. You might have to take a course in microbiology, which isn't really C. elegans, but the point is that there, maybe there are opportunities for this upskilling, and that's something that I don't know. We've had some conversations, they've fallen through the cracks. So I'm going to keep trying to push it and see what we can do. Maybe there's money. I think there is money for it somewhere. Some of the board of trustees for the foundation are interested in this sort of thing, but we haven't really been able to make anything happen in that area. So we'll see. We'll see how that goes.
Just a note that I've continuously had to upscale.
Yeah.
It's feels like my entire life.
Yeah. That's true. And I mean, a lot of people have, it's always learning new skills. It's good, but it's also something that requires some commitment. And it's not so much that you just have to do it. It's sometimes very practical considerations get in the way. Can I take a course at night, or do I really need to take an entire course to learn exactly what I want to know for my thing? So yeah, I think that's always a thing. I mean, that's where a lot of the they have these micro credentials online where you can take, this is common in computer programming, where you can take a micro credential for a language, some developing mobile apps or something, and you can get a certificate for it. But then it's there are other skills that those things just don't exist. So it's interesting. I mean, I think OpenWorm in general could offer a lot in that area, but I don't exactly know what those credentials would look like. So, okay. Well, that's good. So yeah, we can talk about that more. I'll let you know how that goes. So I have this. Oh, here a comment. Yeah, Jesse said looks good. Thank you. So I wanted to talk about one more thing before I get to papers, and this is this thing that Stephen Wolfram published this week, and I don't know how many of you are into complexity theory, but I know some of you know Stephen Wolfram's book, New Kind of Science, which is a book that was published about 20 years ago, and we're kind of on the 20th anniversary of it now. And this is a book that he wrote where he took this tool called a cellular automata. We've talked about this a lot in the group. And he used it to model, just to run these models. And he extracted what he called these rules of order. So like the idea is you have like these grids of cellular automata. And they generate rules if you run them under certain conditions. And those rules then can be classified, so you can like give them numbers. And those rules map to things, like complexity classes in computer science, or sometimes they map to systems like biological systems or physical systems in the real world. So you can build these models that are pretty abstract, that just have a bunch of cells interacting and changing their state. And they can actually approximate real behaviors. First of all, you can build these rules that allow you to then build larger things, from the rules. And one example of that, of course, is morphogenesis. There's a type of morphogenesis you can achieve with these rules, with some of these rules, but also you can map them to more complex systems. And not just developmental systems where there's pattern formation, but emergent phenomena such as like urban settlement patterns or traffic patterns or other things. So there's this idea of the computational equivalence principle, which is that, when I said before that models are pretty abstract and, they, you can't necessarily say that you're modeling something just because you're applying a model that looks like it's similar. You really have to work at getting it, specified in the right ways. There's also this rule called the, computational equivalence principle, which is the idea that you can indeed map from models to real world phenomena, that there is this sort of continuity, that what you're modeling isn't just this heuristic, but it actually does have, some credibility in terms of like modeling real phenomena. So, you have these computational rules of assembly. Those things, are actually what you see in nature. It's just much more abstract than like if you're, watching a bunch of birds flocking, at dusk. That has some computational principles underlying it. So that's the idea here. And he's really into rules. So he has this thing, he says charting a course for complexity, metamodeling, ruleology, and more. So in the world is metamodeling and ruleology. So this is actually a pretty good article. He's talking about in the 1980s, he started working on this. He studied the approaches people tried to use, non-equilibrium thermodynamics, synergetics, which is a type of complexity theory. It's a type of dynamical systems theory, nonlinear dynamics, cybernetics, general systems theory. So we've talked about cybernetics a lot. General systems theory is like a lot of boxes and arrows where you're looking at the flows in a network or in a complex system, and you're trying to characterize the different boxes as entities and the arrows as flows. I imagine that the key question was, starting from disorder and randomness, how could spontaneous self-organization occur to produce the complexity we see? So he assumed that complexity must be created as a kind of filtering of ubiquitous thermodynamic-like randomness in the world. So you have this randomness going on as sort of the initial state. And then you build from there. There's this selection mechanism that selects out a lot of this extraneous disorder, and you end up with order. And so you're looking at things like snowflakes, galaxies, life forms, turbulence. These are all different scales, but they have these things underlying them. And in this case, he's assuming that it's a thermodynamic thing that they share. So they all share this thermodynamic process, this randomness. So this is a very statistical mechanics view of this process. If you were to take someone who knew nothing about statistical mechanics and ask them this question, they might come up with a different answer. But this is his approach. So he didn't get very far at first, but then he started to build his own computer language, and then he started to work with cellular automata, and then he built these programs that would spontaneously generate all sorts of complex patterns. So he's kind of moving forward in this way. He's kind of moving towards, what can you do with these rules now that you have them? So he talks about something here. I'm going to go down through the article. I don't know if there's this metamodeling is this first thing that he talks about. And that is the meta-science of finding minimal models for models. And I know Jesse's interested in this. So but the idea here is this. I was at a complexity conference once, talking to someone who was modeling fish and their behavior. Proudly, the person showed me a simulated fish tank. How many parameters does this involve? I asked. About 90, he said. My gosh, I said, with that many parameters, you could put an elephant in your fish tank too. So that's 90 is not much compared to some of the deep learning models. So this is this is an older context. But if you wanted to make a simulated fish tank display just for people to watch, then having all those parameters might be just fine. But it's not so helpful if one wants to understand the science of fish. The fish have different shapes. They swim around different configuration. What are the core things that we need to see to understand the sort of the phenomenon there? So we need to find the essence. And that essence is usually a smaller number of parameters. But nevertheless, we don't know that. Maybe we're interested in a lot of detail in the model. Maybe we want to know a lot of the variations. So depending on that, we need to have greater or fewer parameters, different parameters, and so forth. But this is kind of getting into this idea of meta-modeling, finding a minimal model for models. So you want to understand how many parameters you need a minimal model of your model, and then you can find that answer. The other thing is this rulology, which is another thing that's kind of odd, and I think rulology is it sort of builds off that idea of rules. So it's the pure basic science of what simple rules do. So that seems kind of like another tautology, but it's actually we have these rules that we can extract from these programs. Now, what happens when we put them together, or what do they actually do? They're generating a pattern. So sometimes if you look at some of these computational rules that Wolfram has produced, they look like things in the world. For example, I think Rule 30 looks like a seashell. All right, the seashell pattern. I don't have a seashell here in front of me, but it's like a pattern of a seashell. It looks certainly like something that would be produced by nature, but it isn't necessarily nature. It's just that pattern. So we can understand the pattern, how this program generated the rule, but we can't necessarily understand, and we can understand how the pattern is generated in the biological system, but we can't necessarily understand how those things connect. They're probably not the same process, but they must have some similarity because they look very similar. So what is that connection there? That's a rulology, at least to me, seems like it's useful for. And so this, I don't know, give you the link to this article. It's fairly long, but it's off of his personal blog. I think that some of you will find it quite interesting, but I would recommend reading it if you want to understand, just kind of ruminate about some of these ideas of complexity theory and biological systems and, that sort of thing. So, okay. Now I'm going to get on with our papers, and if you need to leave at the top of the hour, that's fine. I'm going to go through a couple things here. So we've got a lot of things sort of backed up in this folder here. The first one I wanted to talk about is graph processing systems. So this is something that I think that Krishna said he was interested in at one time, and we might revisit this with respect to some of our posters for NeuroMatch. But so there's this idea that I know some of you have heard of graph learning. So there's an area of deep learning and machine learning called graph learning, and this is an idea that you can learn things by building graphs and representing your data with graphs and then selecting the graphs that are the best. Now, there's also concurrently, there's this idea of complex networks which is where you can model a system with a network and you can analyze the network, or you can actually generate networks from data. And so that's another area that I think I covered a paper on that in my other group a while back. But this is I think it's an ACM article, yeah, Communications of the ACM, and this is the future is big graphs, a community view on graph processing systems. So again, we're kind of revisiting what Wolfram's doing, and actually Wolfram has moved into an area called hypergraphs. So there's this area called hypergraphs, which is like this idea that you have graphs with nodes that are very complex.
So is this the same thing as graph theory, or like adjacent to it? Or?
I think they're building off of graph theory. So like everything that I mentioned builds off of graph theory. So like complex networks are one aspect of graph theory, and it has a history in like social networks and things like that. And then graph learning, I think, is an offshoot of like just, using graphs in computer science. So like graph theory and computer science, and you're just applying algorithms to it. Now, those are graphs, and there are different rules. You can have directed graphs, bidirectional graphs. Now, this is what they're talking graph processing systems. Now, this is where this comes together, because you have this tool, this mathematical tool that then you can use, and it's very computationally friendly. It's not like something you would find with like with a really abstract model you can't represent on a computer. So graphs are by nature unifying abstractions that can leverage interconnectedness to represent, explore, predict, and explain phenomena. Okay, so we have this. It's computationally well understood, and then they kind of get into this sort of idea of a joint effort by computer systems and data management communities. So they kind of get into the history of some of this. They kind of get into what people are doing with them. This is a complex data pipeline for graph processing. So they have, you can build you can extract graphs out of data. You can put existing graph data into this pipeline where you can then analyze them and use them for a number of different, or render graphs in different ways. So it's for scientific computing, for augmented reality and visualization, for machine learning, and you get this processed output. And so this is just kind of goes through sort of the history of graphs and graph processing. And then the next decade, which I think is where they're going with this, with a vision for, applying these types of things. So the idea is, it comes from graph theory. It comes up through a number of different fields. And then the idea is that you have these diverse sources of data, these diverse types of graphs coming together, and that you can process all these different types of graphs in one place, in one pipeline. So I think this is a really interesting thing, and I picked this out. This isn't like graph processing like you might do it in applying it to some specific, data set. This is where you're really kind of pulling together everything in your so like we have different types of data in developmental biology. Even just pick one domain. You have like gene regulatory networks. You have morphological data. You have metabolic data. All of those can be represented with graphs, but they all look very different. They all have different types of nodes and specifications. This is a way to like bring that together into one single sort of representation and then process it. I think it's interesting. So there's that, and then there's this other article on large-scale graph processing systems, a survey. So this is about graph as a significant data structure. It describes a relationship between entities. It kind of goes through a lot of the it is inefficient to use general purpose platforms for graph applications. In this survey, we systematically categorize the graph workloads and applications and provide a detailed review of the existing graph processing platforms, dividing them into general purpose and specialized systems. So this is, again, this kind of goes over this idea of a graph processing system. Okay. Now I want to maybe get into this paper here. Actually, I could do this one. So go into Drosophila gastrulation. So this is a movie here from the paper, and this is Drosophila gastrulation. These are cells, of course, and this is a Drosophila embryo. So at the top is the anterior end, the bottom is the posterior end. And you can see this process of the cells are moving, kind of, they're migrating, but they're migrating in concert. So you can see that they're labeled by colors here, and you can see the movement if it plays again. And then you have the cells that are sort of black and white, and you can see transparent, and you can see the movement of different parts of the embryo. And they're moving in concert. And there's this process called gastrulation where there's this, they're not only migrating, they're sort of folding in different, layers of cells that are moving in concert. So that's the idea, that's what we're trying to understand here in this paper. So this is deconstructing gastrulation at the single cell level. And so the summary of this paper is that gastrulation movements in all animal embryos start with regulated deformations of patterned epithelial sheets. Current studies of gastrulation use a wide range of model organisms and emphasize either large-scale tissue processes or dynamics of individual cells and cell groups. Here we take a step towards bridging these complementary strategies and deconstruct early stages of gastrulation in the entire Drosophila embryo, where transcriptional patterns in the blastoderm give rise to region-specific cell behaviors. So this is where you have these, in the blastoderm, which is this stage of development, you have these transcriptional patterns in individual cells where genes are being expressed, and they're being expressed in a certain way. And this gives rise to region-specific cell behaviors. So in cells in different parts of the embryo start getting these differential transcriptional patterns, or they start expressing differential transcriptional patterns. And this could be like any one gene being regulated differentially in different parts of the embryo. And this is when this starts to happen. Our approach relies on an integrative computational framework for cell segmentation and tracking, and on efficient algorithms for event detection. So what they're doing is they segment the cells, they track the cells to see where they go in space, and then they're able to detect events. And these events could be different genes that are being expressed. So they use these fluorescent markers, and that's why they have the color coding in that figure. And they look at the intensity of it. So it's like the brightness of that fluorescent marker. And you can use like what they call a next-gen technique, which is where you label the cells, and then you put them through a system where you can measure, the level of your target RNA, and you can see how much is being expressed, and that gives you an idea of what the expression level is in a cell. And so you can do this, you can put all this data together to understand this process. So our results reveal how thousands of cell shape changes, divisions, and intercalations, which are like folds, drive large-scale deformations of the patterned blastoderm, setting the stage for systems-level dissection of a pivotal step in animal development. So this is it really kind of goes through all of this. So they have this method for kind of looking at this process. They're able to start, and actually this is interesting because they actually start by forming a two-dimensional projection of the apical site of the epithelium sheet for each time point. Then the cells in the projection of the first time point are segmented and assigned a unique track ID. Cell boundaries and track IDs are then propagated iteratively and automatically over time. They deform each projected image to accurately match the position of each cell within the same cell at the next time point. So they use an algorithm to align the different views of this embryo as the cells are moving around. And it's not just the cells that are migrating, it's that the sheet that these cells form is folding. So you have to understand, you have to have a way to sort of account for that deformation. Once cell positions are matched, cell boundaries, track IDs are copied to the next time point, and then fine-tuned using the watershed transformation. So these are all image processing techniques. Lastly, the segmented and tracked 2D projection at each time point is used to generate a 3D polygonal mesh that reveals the apical surfaces of the cells and an in-toto map of cell-cell adjacencies. And so this is their technique for sort of visualizing all of these data, bringing them together. And so, so they focus on the germ band, a domain of the embryo that converges and extends as a consequence of multiple cell intercalations. Most of these events involve cell quartets, so cells in four behaving together, and proceed through so-called T1 transition, in which two non-adjacent cells come together and split the orthogonal cell pair. Another intercalary event involves more than four cells and proceeds through a configuration where several contracting interfaces generate a so-called rosette state. So these are like these clusters that they form. So they are able to look at all these different types of geometric transformations that the cells are making as they're moving around and they're engaging in these intercalations. So this is a very good way, they've kind of worked a lot of this out algorithmically, so they have a bunch of criteria that they use. Let's see if they have any nice figures in here. I'm sure at the end here they do. Okay, so this is exactly what it looks like. This is the Drosophila embryo. It's different than the C. elegans embryo because there are a lot more cells, but also they go through this state of cellularization. So they have these different, C. elegans embryo, the cells are pretty much divide and they sort of form this, what it's going to look like in the adult with some, there's some differentiation in the embryo, but then, it's not that the developmental states, you don't have these transformative states like you do in Drosophila. In Drosophila, you have the state where there's cellularization, and then there's a lot of folding that goes on here. So you have these different parts of the embryo that fold together, they fold over, and they turn, and they do a lot of coordinated movements. So this is one of the main features of its embryogenesis. And so then they can take these cells and classify them. They have invaginating cells and cells that remain on the surface, and they can look at those. They can also look at some of these things in this ventral view in this Mercator projection, which is taking this curved surface and flattening it out. And this is, of course, we've talked about this with the axolotl data, where we're trying to take flat images and we're trying to wrap them around a sphere. This is the opposite process, where you're taking this, I don't know if it's a sphere, it's like an elongated bean, and flattening it out. So this is a similar problem. So if this is something that might be interesting in that respect. And then they have this dynamics of cell intercalations. So you have these, what they call a T1 transition, that kind of show what that looks like in this 6-cell rosette over time. And they show what that looks like. So then they show this in a diagram here with like a two-dimensional diagram where they show like how these cells move with respect to a central cell and its surrounding cells. So that's all for that paper. And I think probably, let's see, one more thing. Whoa, I don't think I've ever shown you this. So this is something that I created for one of the talks, and this isn't the right one though. Oh, these are sketches. So I have I've mocked this up a lot better since then. But this is an example of a hypergraph. So this is an example of something that looks like a C. elegans embryo, but it's actually like this idea of a divergent generative integration hypergraph that you would see in like a C. elegans like embryo. So I drew this out one day, and I was sitting there, and I did it in pencil first, and then I kind of worked out some of the details on this later. So this is kind of an early draft of what this looks like, but this is good enough. I can explain what this looks like. So you start with a single cell, and you go to like a 4-cell embryo, which doesn't really have much differentiation. When you get to the 8-cell embryo, you get a germ cell. So that splits you into two networks. The germline can form a network, but it's not really, there's not much there. It's just kind of like germ cells. But they're segregated out from this other network, which is the somatic network. So the point is that there's a division point here in terms of your networks. Now each one of these nodes represents a number of cells that form a network. So this is what they call a hypergraph. So this is 7 cells here. This is actually a network within this node. This is 14 cells. This is 28 cells. And the germline continues to increase, but in a different network. So they don't interact at all. Then when you get to 28 cells here in this putative organism, this splits off. So now you start to get neural cells that start to differentiate. But you still have your embryo network. So that network expands in size as well because you have cell divisions. So now you have a network of 55 developmental cells or somatic cells and 3 neural cells, and they form a very small connectome. But then you begin to start, you start getting more and more neurons, and you continue to see more somatic cells, and you get this interesting phenomena where you can get cells that at one stage were part of the somatic network or this embryo network that then differentiate into neurons and join this network. So they actually switch to a different subnetwork. And so you can see there isn't any interchange between the connectome and the embryo network. It's just in this direction from the embryo network to the neural connectome. And so you can see that over time and then when you get to 194 cells in this embryo network, you get a further bifurcation, which is basically where you're starting to get different parts of the nervous system. So in this case, it would be in the head and maybe in the tail of C. elegans or whatever this organism looks like. And then you get interchange between those networks, perhaps, and you get defections to the connectome. So this is what this hypergraph looks like. And I've kind of worked this out since then. The idea is that it's like a lineage tree, but it's actually a set of networks that differentiate over time. And so you can characterize the entire embryo with a series of subnetworks. And you can say things about the interactions between them. As those cells diverge, they form divergent networks. But there are also interactions between the networks that are maybe more subtle. That's the idea there. So I think that's it for today. I think Susan's the only one left. Okay.
Everybody had to leave. I thought I had to leave. I had something at 10, but I have no idea what it was. So
okay.
Yeah, no, your explanation of the fly there that looks interesting. I should get that paper because that fits in with the axolotl images.
Yeah, I think the idea of taking the image and warping it in a from a 2-dimensional, 3-dimensional, or 3-dimensional to 2 dimensions. There's that. Yeah. Doing that well is the best.
Yeah.
It's a hard thing to do, but if you can do it well, then it's very informative.
Yeah. Thanks for showing that paper. That's great. Okay.
All right.
Thank you. Thank you.
No problem. All right. Well, thanks for attending, and next week we'll have more to say about different things. Hacktoberfest will have started, so we'll go over maybe some initial things there, and tomorrow.
Okay, I'd like to see more about that. I might be able to participate, but I don't know anything about it, and my computer programming is a little rusty, and it's just something like that. So if I got to see it, I might be more inclined to participate. Just saying.
Yeah, I will.
Anyway, meanwhile, signal processing and then learning some Python. Anyway.
Yeah. All right. Have a good week.